The technique that I've developed is called SBARRO, which is kind of cheekily named after the airport pizza chain.
Yeah.
It stands for Synaptic Barcode Analysis with a Retrograde Rabies Readout. It's kind of a mouthful, so kind of a cheekily name, helps the medicine go down.
I like the acronym, yeah.
Yeah, and I developed it as a postdoc in Steve McCarroll's lab, which is at Harvard Medical School in the genetics department, and part of the Stanley Center, which is at the Broad Institute. So as a postdoc, I kind of laid the foundation for SBARRO and published the proof of concept work that we could track the infection of individual rabies virus particles. So the way the system works is all synaptic circuits have two layers, right? There's a layer of postsynaptic neurons, which are the listening neurons, and each postsynaptic neuron gets inputs from hundreds or thousands of presynaptic partner cells throughout the brain. And we can consider these the singing neurons, right? And each individual postsynaptic neuron kind of gets a chorus of inputs from its presynaptic cells. And so our strategy using SBARRO is to take thousands of instances of these kind of personal choruses that individual neurons get, and to use them to build these quantitative maps of connectivity. So there's many different cell types in the brain. So if you have a certain cell type, that's the listening neuron, it gets a certain type of chorus of inputs. And then each of the input, the singing neurons, itself has its own chorus that it's listening to. So by breaking apart the neural circuits into these two-stage systems, we try to reconstruct a whole from many sums of the parts.